Welcome, everyone. Good morning and afternoon to everyone. Welcome to today's Visions webinar. My name is Donna Cohen, Director of Events of Western Region for the Foundation Fighting Blindness. We hope everyone is staying healthy and safe. It is my pleasure to welcome you to the first of a series of webinars being offered over the next several weeks, and we are excited that we can continue to provide you with research updates. If anyone has questions that are not answered in today's session, please email them to info@fightingblindness.org. Over the course of the next 2 hours, you will hear from experts in retina research who will provide you with updates on some of the latest technologies, treatments, and research. So now, with further ado, it is my pleasure to introduce Ben Shaverman, Senior Director of Science Outreach and Community Engagement for the Foundation Fighting Blindness. Ben?
Hi, Donna. I am just, and hi, everyone who has joined us. I am just getting my slides set up. Hold on. Thank you. I presume everybody can see my opening slide okay and can hear me okay. And welcome. It's a real honor and a privilege to have you join us today for this update on retinal research. And I hope, as Donna said, everybody is safe and well. I know this is a really unusual time. And for me, I'm usually out in the community at hotels and libraries and other meeting places delivering these retinal research updates. But today, greetings from my kitchen, where I don't do a whole lot of cooking. So it's a real pleasure, again, to be here. Another point I want to make is the Foundation Fighting Blindness, while obviously we raise a lot of money for research, that's our primary mission. I think during these times, we really are family. And I know all of you out there, the research community, constituents, you feel like family to me, and I am glad that you joined us. I will be going through my presentation. That will take about 45 minutes. And then we'll go straight to our panel. And we have a great panel today. I think my talk will be a nice warm up for the real headliners. Dr. Chung, Dr. Park, and Dr. Rodman. You'll get a lot of diverse perspectives from them on the research. I'll be covering today some of the highlights that are going on in gene therapy, stem cells, some other approaches. And there are a lot of things I won't be able to cover. There's just too much going on, and that's actually good news. But as you've heard, you can email, you can ask questions today, but if your question does not get addressed, send those questions to info@fightingblindness.org, and we will get to your question. So I always like to open my presentations with some context for where the research is. And today we're approaching about 40 clinical trials for emerging therapies for inherited retinal diseases and dry AMD. And this is a remarkable achievement, given that for really the first 40 or so years of the foundation, we were founded in 1971, there was very little happening in clinical trials. There was very little human research being done. And another new fact that I've included in this opening contextual slide is that in addition, or including the clinical trials that are underway, there are about 80 or even more than 80 therapies that are in development. That not only includes those that are in clinical trials, but those that are nearing clinical trials, maybe a couple of years away. And they're very diverse. Again, stem cells, gene therapy, small molecules, some target specific genes, others are cross-cutting and will hopefully work for a broad range of people. And I think it's important for everyone to understand that you'll be hearing a lot about different companies that have moved into our space. Remarkably, dozens of companies. And I'm proud to say the Foundation Fighting Blindness played a significant role in getting research to the point where we could attract investments from many of these companies and also lead to the development of these companies that otherwise wouldn't exist. The Sparks, the AGTCs, the Night Stars, many of those would not be around if it wasn't for foundation funding. Our current portfolio is about 80 projects. We fund a variety of research efforts, emerging treatments that are still in academic settings, academic labs. We fund translational projects to move emerging therapies out of the academic labs and into the clinic. We fund some early clinical trial work, career development awards, the Jason Commanders, Rachel Huckfelds, Mark Panisis. It's a very extensive list. These are all relatively young clinicians who we have funded so they could establish their careers in our space. We fund natural history studies, and as you'll be hearing about, we fund a no-cost genetic testing program, as well as the Retina Tracker registry. Always when I think we talk about retinal research, it's important to review what the retina is all about. And that's what you see on the left side of the screen here. It's a very complicated piece of tissue. This is actually a side view of the retina. The actual human retina is only about 0.5 millimeter thick, and it lines the back of the eye. And when light comes into your eye, goes through the cornea and the lens and the middle part, we call them vitreous, it hits the retina, this very thin piece of tissue, and it's the retina that turns the light into signals that our brain interprets as vision. And the retina is a very frail piece of tissue. It's almost like wet tissue paper. And each of our retinas has about 125 million photoreceptors. And those are the actual cells that process light. So there's a lot of power packed into a retina. And in this particular diagram, you can see toward the top, there are these vertically oriented cells, and those are the photoreceptors. The red, blue, and green cells are cones. Cones are concentrated in the center of our retina in an area we call the macula. And there's an even smaller target called the fovea. But anyway, cones are the cells that are most important for our activities of daily living. They enable us to perceive detail, to read, to recognize faces. They give us vision in lighted settings. They give us color perception, and they give us central vision. So again, those are concentrated in the center of the retina. On the periphery, we have rods, and rods give us vision in dark settings and peripheral vision. And another layer of cells that are very important I want to cover before we move on are the retinal pigment epithelial cells. And in this diagram, they're at the top, they're a single layer of cells, and the photoreceptors are nestled into these cells. And the RPE cells provide a support system for the photoreceptors. They provide nutrition and a very critical waste management process. And often in macular conditions, the disease manifests in these RPE cells. And unfortunately, when the RPE cells aren't working correctly or they degenerate, then the photoreceptors degenerate. So on the right side of this diagram, I just have a blow up of a rod cell and a cone cell. You can see they're very long. The top part of the cell is the sensory part that's sensing light. The bottom part of the cell is the typical cell machinery, the engine that's making all the proteins and sending stuff up and down that long sensory portion so that vision is possible. So the first treatment approach I want to cover today is gene therapy. And just a little background, for inherited retinal diseases, RP, Usher syndrome, Stargardt disease, choroideremia, the whole spectrum, people get these diseases because there are mutations in a single gene that in each individual that's causing the disease. The challenge for us is there are 270 of these genes, again, each of which, when mutated, can lead to a retinal disease. But really decades ago, 25, even 30 years ago, researchers envisioned that replacing that mutated gene for everybody would be an elegant way to potentially save and restore vision. And that's what gene replacement therapy, sometimes we call it gene augmentation therapy, is all about. And of those 38 or so clinical trials I talked about, a vast majority of those are for gene replacement therapy. And the real simple way to look at gene therapy is you're replacing that mutated gene with a healthy gene. Now, the way researchers are doing that most commonly today is they're actually using a human engineered virus to deliver the therapeutic gene to the cells in the retina. Unlike the virus that's getting so much attention now, these viruses are human engineered. They don't replicate. They do not do anything bad to the cells. They are a delivery system. And again, the researchers put the therapeutic gene in the virus. These are injected underneath or sometimes in the middle of the eye, underneath the retina or in the middle of the eye, and then they're absorbed by the cells in the retina. And again, so far so good. We have our first FDA-approved gene therapy, which I'll talk about in a moment, and many other gene therapies that are in clinical trials right now. Most, again, are performing well. I don't list everything in this talk, and I will cover some other approaches that use gene therapy and other forms of gene modification and gene editing as we go along here. But first, I want to talk about LUXTURNA. Again, I mentioned we have an FDA-approved gene therapy. LUXTURNA is the first gene therapy approved for the eye or an inherited condition. It was approved more than 2 years ago in December 2017. And this is for people that have mutations in the gene RPE65. And mutations in that gene can cause labor congenital amaurosis, a pretty severe retinal disease that affects kids. And it can also cause RP. So this treatment was really envisioned back again in the mid-90s. The Foundation Fighting Blindness began funding some early research for this to move it into a clinical trial. And that clinical trial began in late 2007. And shortly after that trial began, in a matter of just a few months, the participants, some young adults and then children, reported dramatic improvements in their vision. Some people were able to put away their navigational canes. They were seeing the faces of loved ones for the first time. We even had reports of people seeing stars in the sky for the first time. So this was a very effective gene therapy right out of the gate in the first clinical trials. As these trials moved along and researchers began treating kids as young as 4 and I believe even younger, Children's Hospital of Philadelphia, who was conducting this trial, spun off a company called Spark Therapeutics to raise the money to get it across the finish line and get FDA approval. And again, that happened in December of 2017. And now several dozen people over the past couple of years have received the treatment commercially. And there are centers of excellence around the United States that can deliver this treatment. As a footnote, Spark was recently acquired for more than $4 billion from Roche Pharmaceuticals. So this early trial, which again began in, well, the research actually began back in the mid-90s, has really blossomed into a remarkable treatment. And I think what's equally important as the people who are benefiting from the treatment today is the fact that this trial really paved the way to use gene therapies to treat a variety of other inherited retinal conditions. And that's what we're going to cover in the subsequent slides. A company that was spun out of the University of Oxford in London is called Nightstar. They were recently acquired by Biogen. They have a couple clinical trials underway, one for, and again, gene therapies, one for a condition called choroideremia, which has some similarities to RP. It also not only affects photoreceptors and RPE cells, it affects the outer vasculature of the retina, the choroid. They are now in a phase 3 trial, which is the pivotal trial. If this is successful, Nightstar Biogen will likely go to the either the FDA or the European Medicines Agency, or both, to get regulatory approval. They had some very nice results in their phase 1/2. Most people had some improved or at least maintained visual acuity in that phase 1/2, and we look forward to seeing reports from phase 3. This same company has a gene therapy clinical trial underway for X-linked retinitis pigmentosa caused by mutations in the gene RPGR. Most people who do have X-linked RP have mutations in this gene. It's recently moved into a phase 2/3. In the phase 1/2, they did report some improvements in visual fields and retinal sensitivity, so more encouraging results. And as a side note, the Foundation Fighting Blindness has been pretty active in funding X-linked RP research at both Kellogg, at the University of Michigan, and Ann Arbor, and some really pivotal canine studies at the Vet Center at the University of Pennsylvania. So we're very pleased that that work helped inform the X-linked RP trials. Another company that's reported some very encouraging results as of late is called Applied Genetics Technology Corporation, or AGTC. They too have an X-linked RP gene therapy clinical trial, again for mutations in RPGR. They recently reported that people who had their retinas treated centrally had improved visual acuity at 6 months. People treated peripherally had maintained vision, which is a positive thing. If you can halt progression, that's great. And they're planning to report more of this phase 2 data later in the year, and they're starting to consider a phase 3 if that data continues to be positive. They also have 2 clinical trials underway for a condition called achromatopsia. There are 2 genes that cause, when mutated, cause the majority of achromatopsia cases. That's CNGB3, CNGA3. Those are still in early stages, phase 1, 2, but both in early reports, people with achromatopsia reported better tolerance of bright light. And that's important because people with achromatopsia are extremely light sensitive. It can be even painful to be in bright light. So if you can help improve their tolerance to bright light, that's a very positive thing. Again, these are early trials. Stay tuned for further results later in the year. And I just wanted to provide a list of some other gene therapy and these are all gene replacement clinical trials that are underway. Spark has a phase 1/2 trial in the US. Sanofi has been conducting gene therapy clinical trials for Usher syndrome type 1B and Stargardt disease. They're looking to get a partner for these programs because they're getting out of the ophthalmology space. But stay tuned for progress there and them trying to get some new partners. In Europe, we have a couple of clinical trials for retinitis pigmentosa, PDE6 beta and RLBP1. Those are underway in France and Sweden, respectively. We're very pleased to let you know that Shannon Boy at the University of Florida was able to get her research out of the lab and into a clinical trial at the University of Pennsylvania. That too is for LCA, but that gene is for a gene called GUCY2D. We also call that LCA1. That trial is just enrolling patients now. And again, we provided some significant funding to her. And one of our partners, Mira GTX, is also in the achromatopsia and XLRP gene therapy space. They have not reported any real meaningful results yet, but stay tuned for more reports of progress from them. So I'd like to shift gears from gene replacement therapy to CRISPR. And I'm sure many of you have heard about CRISPR-Cas9 in the media, gene editing. And it's the basic concept here is instead of replacing an entire gene for a patient, delivering a whole new gene to them, you're going in and you're providing a pair of molecular scissors, if you will, to cut out and potentially replace the mutated portion of the gene. And where this can come in very handy is when the gene is difficult to deliver because it's big. I talked about those viruses that are being used to deliver the whole gene. Well, they have capacity limitations, at least they currently do. So another way to approach that challenge is through gene editing. Again, just editing the gene instead of replacing it. And collaboration between Editas and Allergan recently launched a phase 1/2 clinical trial for this approach for a mutation in the gene CEP290, which is a big gene. When it's mutated, it causes a form of LCA. This is just getting off the ground. They're just starting to administer the treatment to patients. And also on the radar screen is people with USH2A caused by a mutation in a region of that gene called exon 13. So hopefully that will move into a clinical trial before too long. So this is actually editing the gene, the DNA. But there's another approach, and it is sort of a form of editing, but instead of editing the gene, you're editing the messaging system within the cell. So every cell has a complete set of DNA, and that information, that DNA information, is communicated in the cell through something called messenger RNA. And if there's a mutation, the mutation is also transported in the messenger RNA. So ProQR, a company in the Netherlands, has come up with an approach to address the mutation in RNA. And that approach is, it's a long, complicated word. It's called an antisense oligonucleotide, or AON. And basically the way this approach works is it masks, it sort of covers up the mutated part of the RNA. So when the cell gets the message, it can make the correct proteins. That's what cells do with DNA and RNA, is they use that information to make proteins. So you can think of antisense oligonucleotides, or AONs, as being like whiteout. For those of you that have used typewriters, you're old enough to have that experience, and you used whiteout to mask a word. This sort of, in a very simple sense, works like that. The Foundation Fighting Blindness is investing up to $7.5 million in an approach for USH2A, again, mutations in exon 13. But that trial, we expect to hear some reports from that in the not too distant future. But prior to that trial, ProQR launched a phase 1/2 trial, again for CEP290, for a mutation in that gene, and had some very impressive results. About 60% of people in that trial had improved visual acuity, as well as improvement in mobility and in dark settings. Really stellar results for an early phase 1/2 clinical trial. So that trial has moved into a phase 2/3. And ProQR is also working on an approach, antisense oligonucleotide, for people with P23H rhodopsin mutations, which can cause RP. And also in development, they're focused on Stargardt disease, another mutation in USH2A, and they're also looking at some undisclosed forms of LCA. So ProQR, really out of the gate, had very excellent results, and their portfolio has expanded very impressively. And what's nice about this approach, it's something you can modulate. It's not a one-time treatment. It's something that's delivered perhaps a few times a year. We can have Dr. Rodman elaborate more on that when we get to the panel. But it's not a one-time thing, and that can be beneficial if you want to modulate the dosing. So we're going to shift gears again when we're talking about gene therapy. And this is getting back to delivering an entire gene to the retina. But instead of delivering a healthy gene to replace the bad gene, this is delivering a gene to harness different cells in the retina for vision. And this approach is called optogenetics. So on the diagram here, I have this ominous X that's going through the photoreceptors. And the reason I've done that is to signify somebody with advanced vision loss, whether it's RP, AMD, whatever condition. And by definition, if you've lost all your photoreceptors, you don't have vision, because those are the cells that make vision possible. And replacing a gene, the mutated gene, is not going to make sense because you don't have any photoreceptors left to save. But what researchers figured out in lab studies not all that long ago is that there are these other cells in the retina, ganglion cells and even bipolar cells, that are lower down in the diagram. I have a big oval around those. Those cells survive after the photoreceptors are gone. And in lab studies, they delivered a gene to these cells that makes them light sensitive. And they operate somewhat like photoreceptors. So optogenetics is a way to harness other cells in the retina, cells other than photoreceptors, to restore vision. And what's really exciting about this approach is it can potentially benefit people with the most advanced vision loss, and it's designed to work independent of whatever gene mutation is causing the disease. And there are now 3 clinical trials underway. I see I updated the bullets in my trial slide here, but I did not put 3 instead of 2. Anyway, there are 3 trials that have begun for this approach. Allergan, which has had a trial actually underway for a couple of years here in the US, maybe even longer. GenSight, a UK company who's studying this approach in humans in France. And then recently, a company called Bionic Sight, in partnership with AGTC, is conducting a trial for this in Long Island, New York. That trial is just off the ground. They have not publicized it much, but it is enrolling at this point. And some of these approaches, in addition to treating the actual retina with this optogenetic therapy, there are glasses or goggles that enhance the information that are coming into the retina to hopefully give a better visual experience for the user. A cautionary note here is we're still not sure exactly what kind of vision optogenetics is going to restore. We're very encouraged by what was observed in animal models, but I think we have to wait and see how these trials pan out in humans before we get too excited. And I will make a comment that the Foundation Fighting Blindness is also funding John Flannery at UC Berkeley for sort of a second generation version of optogenetics. And you should be hearing more about that in the coming months. So that concludes my overview of gene-based therapies. And let's get into a relatively brief overview of stem cells. So the real power of stem cells is that you can use them to grow basically any tissue in the retina, whether you need photoreceptors or RPE cells or both. And you can use these cells to either protect existing cells in the retina or to try to replace cells that have been lost to advanced disease. And you'll be hearing more about stem cell therapies from Dr. Park, because she's done some very interesting research in this space. And there are a lot of sources that you can get stem cells from. Dr. Park gets them from bone marrow, but also one of the researchers that we've been funding for several years, David Gam, is deriving stem cells from adult blood or skin. Those are called induced pluripotent stem cells. And what he can do is he can take a cell sample, a skin sample, a blood sample from a healthy person, genetically tweak those cells so those cells revert back to a stem cell-like state, and then he can coax them forward to become retina. So healthy adults can now be stem cell sources, patients or even people without retinal disease. So there have been really exciting advancements over the past decade in this space. Again, David Gam's work is very exciting, and I urge you to stay tuned to what he's doing. In the meantime, I wanted to cover a few clinical trials that are underway, and the first two use a stem cell type called a retinal progenitor. And this is a stem cell that is almost developed into a photoreceptor. And in this first trial that's been launched by a company called J-Site, the idea is you inject these cells into the middle of the eye, into the vitreous, and the goal, the hope, is that these cells help keep existing retinal cells alive. And we call this neuroprotection. And J-Site has this in a clinical trial. It's a phase 2B. They're targeting people with RP, Usher syndrome, and some other retinal conditions. RP and Usher, I think, are the main conditions at this point. This came out of the lab of Henry Clausen at UC Irvine. In the phase 1-2 portion of this trial, 28 patients were treated, and they saw that the patients in their treated eyes, those eyes performed better than the untreated eyes. And all of those patients, they treated one eye and compared it to the untreated eye. So now they've moved this trial into a phase 2B. 85 people are enrolled, and we hope to hear results before too long to see how well this is performed in a larger population, and actually a higher dose, and for people with better vision. So stay tuned. Now on the next slide, a company called Regeneron, actually they're in the UK, is using a very similar cell type, a retinal progenitor, a stem cell that has almost become a photoreceptor, but instead of trying to just protect cells, they're trying to replace cells that have been lost. And their trial is, again, for people with RP, Usher syndrome, and potentially related conditions. It's underway at Mass Eye and Ear and a practice in Phoenix. In their phase 2, about a year ago, they began reporting that patients had improvements in vision on average, about 3 lines, and those improvements have extended out to 12 months in a phase 2. So again, encouraging results. Stay tuned as this moves forward in the clinic. Leading this trial is Jason Commander. Early on in my talk here, I talked about career development awards. We've provided Jason with a lot of career development funding, and I'm very pleased to say he's turned out to be quite the clinical researcher. And this project in general, not only have we funded Jason, but we've provided significant funding to the lab studies that made this trial possible. And then in the age-related macular degeneration space, there are many trials that are underway using various forms of stem cells. As I mentioned early on in the talk, people with AMD, their disease manifests in RPE cells. So a number of companies are coming up with ways to replace those RPE cells. And in the case of a company in Southern California called Regenerative Patch Technologies, they're actually putting RPE cells on a scaffolding, this very thin polymer, so that when the cells are implanted in the retina, they're properly oriented, and they believe the scaffolding will improve their chances of survival. It's been a while. They reported some early results. Those results were encouraging, and we're waiting to see, or at least hear about more results from them in the not too distant future. Project called the London Cure for Blindness project, underway in the UK, is using a similar approach for people with wet AMD. There are these types of studies underway in Japan, and then there are many international companies that are transplanting RPE cells without the scaffolding. So stay tuned for these. And I just have about 2 more slides on emerging therapies that I wanted to cover. And we're moving into, again, a different approach, therapeutically speaking. And these are cross-cutting therapies, both major foundation investments. And both of these are gene-independent approaches that are designed to potentially save the retina, really neuroprotection. And one company that we're funding and we helped get off the ground for their approach is Nacuity in Dallas. And we're investing up to $7.5 million in this approach. And through research at Hopkins that we also supported, they've come up with a very strong antioxidant called NACA. That's the short name, N-acetylcysteine amide, for those of you keeping score. But NACA showed really nice results in lab studies for slowing vision loss in RP and potentially other conditions. In retinal disease, RP, AMD, most retinal diseases, oxidative stress has been shown to exacerbate vision loss. The cells are not effectively processing the byproducts that come from oxygen processing. And so this will be moved into a clinical trial here in the United States, hopefully within the next year. They're actually doing some clinical research in Australia for this. It would be an oral medication that people would take. Again, because it's something taken orally, you can easily modulate it, and it's designed to work independent of the mutated gene causing the disease. We're also funding a project in France being developed by a company called Sparing Vision. That investment is up to 7 million euros. And the idea here is to deliver a protein that helps keep specifically cones in the retina healthy. Now, for those of you with RP, you may know that RP usually manifests in rod cells. And that's why people with RP lose night vision and peripheral vision. But what happens over time is as many of the rod cells degenerate, the cone cells degenerate. And some very smart researchers in France, José Sahel and his colleagues, discovered a protein that rods produce and secrete that keeps cones healthy. And they aptly labeled this protein rod-derived cone viability factor. And they've developed a therapy. It's a one-time injection that is designed to deliver that protein to keep cones healthy. Again, this was developed in Paris at the Institut de la Vision. We funded a lot of early research that made this possible, in addition to the current work to move it into a clinical trial. And they're hoping to get this in humans in 2021. And then finally, I just wanted to talk about some Stargardt disease therapies that are in the clinic. And I'll start off by noting this is just a subset of things that are in or moving toward the clinic for Stargardt disease. And to briefly go over this list, I wanted to emphasize the fact that Stargardt disease is a waste accumulation problem in the RPE cells. When vitamin A is metabolized, there's waste that accumulates in the RPE cells of people with Stargardt disease. That's what happens because of the gene mutation. So these companies are coming up with ways to modify or alter vitamin A processing so there's less waste in the RPE cells. Alkeus has a phase 2 trial for a modified form of vitamin A that quote unquote burns cleaner than the vitamin A that we get from our diet. So the approach there is you eat a diet that's kind of light on vitamin A, you take this vitamin A, and the hope is you'll have less degeneration in your RPE cells. Companies like Acucella and Belite, they're actually trying to slow down the uptake of vitamin A into the retina. We call this modulation, vitamin A modulation. The Acucella trial is actually, in a phase 3 at this point. A company called Belite is planning to move their approach into a phase 2 trial soon. Then finally, there's another company called Iveric Bio, which actually has a very diverse portfolio. One of their products, emerging therapies called Zimura, is targeting the complement system to try to slow down Stargardt disease. That's a little different from targeting the vitamin A metabolism process. So the complement system is a part of our immune system that fights off harmful bacteria and other foreign invaders. But in people with retinal diseases, especially macular conditions, the complement system is overactive. So companies are trying to kind of temper the complement system to slow degeneration. And that's what Iveric's emerging product, Zimura, is designed to do. But in addition to this drug or protein, if you will, that targets the complement system, Iveric has gotten into the gene therapy space. And they're actually trying to target gene replacement therapies for some of the challenging conditions, including Stargardt disease, caused by ABCA4 mutations, rhodopsin, which is a cause of RP when that gene is mutated, and Best disease. These are all still in a preclinical mode, but they are working to move those into clinical trials. Okay, we're done with therapies, and I just want to talk about our no-cost genetic testing program briefly. So it's very important for people with the inherited retinal conditions to know what mutated gene is causing their disease. As I said, in just about all cases, it's one gene. And if you can figure out what that gene is, you're getting to the root cause of the condition and you're getting a definitive diagnosis. And so there are other benefits to genetic testing. It helps you understand the inheritance pattern within your family, and perhaps even more excitingly or equally exciting for people is it can help you qualify for clinical trials. So in a vast majority of cases, people with inherited retinal diseases really benefit from genetic testing. And the Foundation Fighting Blindness is offering no-cost genetic testing through a partnership with Blueprint Genetics, which includes genetic counseling from Informed DNA. And now, any patient with an IRD, as long as you haven't had broad panel testing in the past 4 or so years, any patient can go to their eye doctor, optometrist, ophthalmologist, retina doctor, and their doctor can order the Retina Tracker genetic testing panel from Blueprint Genetics online. Their panel, and I have to update my slide, I see another correction I need to make. Their panel is up to actually 322 genes. It includes some mitochondrial genes, which are also associated with eye and retinal diseases. Anyway, their panel is outstanding. There's no other panel really in our space that goes more broadly or deeply. It includes screening for RPGR, which is a major cause of X-linked RP, and it can detect other harder to find mutations, copy number variants, and intronic mutations. Another very important aspect of this program is that your privacy is always protected. We never share your personal information. With other programs that are offering no-cost genetic testing, that may not be the case. So if you're considering genetic testing, we recommend that you talk about the privacy and whether your personal information may be released. Anybody who gets genetically tested through our program needs to get genetic counseling. Informed DNA is a great group. If the facility wants to use their own genetic counselor, that's fine as well. And as our genetic testing process has gotten off the ground, we are now reporting results in less than 2 to 3 months or more at 4 to 6 weeks. So that process has gotten streamlined as well. And finally, I wanted to implore everybody to put their disease information in our patient registry called My Retina Tracker. And this has actually been around for about 6 years. And what we need to remember for people with inherited retinal diseases is their information, their diseases are rare. There isn't a whole lot of publicly available human data, or at least data available to the research community on these conditions. And that's why we launched My Retina Tracker, so that patients and families can upload their disease information. If they know their genetic mutation, great. If they don't, that's okay too. But we make this information available to the research community, including companies and researchers recruiting for clinical trials. The registry is available at myretinatracker.org. We have more than 15,000 active registrants. And through My Retina Tracker, we've traditionally offered, through the registry, offered genetic testing. Now you don't have to be in the registry to get the genetic testing. We hope you will go into the registry. We strongly recommend it, but that's no longer a requirement. And it's really a great way to get on the radar screen of the research and clinical community. And another fact about the registry I wanted to share is that over the past 5 to 6 years, we've gotten queries now from more than 150 researchers and companies looking at the data and doing clinical trial recruitment. So we hope you will get into My Retina Tracker. Stay tuned as we're launching a new version of the registry in another month or two, and you will be hearing more about that as we get closer to that launch date. And then finally, a little shameless self-promotion, but maybe an activity for you or your kids while you're isolated in your homes. Over the years, with the Foundation Fighting Blindness, I've had the privilege and the pleasure of interviewing kids and families that are affected by retinal disease. I've been greatly inspired by their courage and their enthusiasm and the cool things they do. So that inspired me to write a sci-fi young adult novel called Retina Boy. And as I say here, it's a story of friends, aliens, retinas, and rock and roll. It's about a kid in Iowa who's born without retinas. The researchers are mystified. They've never seen a condition like this. And as it turns out, not to give you too much of a spoiler, but he's actually an alien from another planet of blind inhabitants. And the story takes off from there. But he grows up to be an accomplished rock guitarist. He has a band. His girlfriend is the smartest kid in the class. She happens to use a wheelchair because she can't walk. And it's a lot of fun. I recommend it for people about 10 to 12 or older, very appropriate for adults and kids. It was published by Apprentice House, which is Loyola University of Maryland. It's available in many forms on Amazon. And for a limited time, I am giving away copies of the Audible and Kindle version. And you can request a version while I still have these versions available by sending an email to bshaberman@fightingblindness.org. So I've gone a little further in time than I wanted to go, but that concludes my presentation. Oh, one more slide I wanted to give everybody before we move to the panel. Please make it a point to visit fightingblindness.org for updates and research. We have a new article from David Gamm that'll go up next week. Clinicaltrials.gov is a great way to learn about all the clinical trials that are underway in addition to fightingblindness.org. And again, make sure you register in my Retina Tracker at myretinatracker.org. So that finishes off my presentation. And I think we can move on to the panel. If we want to switch screens and go to our panel slide. Do I have to release my screen?
There's one thing.
So Chris, can you share that panel screen? Great. There we go. Well, I'm always very pleased and privileged to moderate these panels because this is where we have the researchers, both clinical and lab researchers that are really doing the heavy lifting on the front lines. And we have a really great group with us today. They include Dan Chung with Spark Therapeutics. He's been very involved in the LUXTURNA trials, but I will caution, I know because of regulatory issues, now that you have an FDA-approved therapy, you can't talk about the LUXTURNA trials in a lot of detail. But I know Dan was involved in developing the mobility course that that was used as an outcome measure to help show that LUXTURNA was actually working. And then we have Dr. Susanna Soonchun Park from University of California, Davis. And I've been in touch with her over the years, hearing about her bone marrow stem cell clinical trial. So I'm excited to hear updates about that. And then last but by no means least is David Rodman, who is with ProQR Therapeutics, and all those great trials that I talked about for LCA, USH2A, rhodopsin, that's what they're involved in, and I'm sure he'll be happy to elaborate on those more. But to get things going, I'm going to go through a couple of questions with the panel, and then we'll open it up to questions from those of you that have joined the webinar. But to start off, Dan, Dr. Chung, can you talk a little bit about your role at Spark?
Sure. Well, it's definitely a pleasure to be with you all today, and thanks to Ben, Donna, and Chris for some fast adjustments from a live meeting to this virtual meeting. So for myself, in this time, I've been homebound now for about 3 weeks, so it's good to join you all virtually. I trained as a pediatric ophthalmologist with extra training in inherited retinal disease and retinal gene therapy. I serve as Spark's ophthalmologist and IRD resource based in medical affairs, heavily involved in clinical development and pretty much any other facet of the company that needs ophthalmology expertise. So one of those things I get to be involved in is patient advocacy, which I very much enjoy. So I do a little bit about everything. And as Ben mentioned, I've been at pretty much every level of development for Regenerative, which is the RP65 gene therapy at University of Pennsylvania Children's Hospital before I came to Spark. And then obviously the post-marketing and approvals that we received once we had Spark developed.
Great. And Regenerative is LUXTURNA. Are you able to say LUXTURNA, Dan?
I think that I might be able to.
I don't mean to put you on the hot seat there.
Yeah. I think because of the virtual world, I don't know exactly who's out there. So, but Regenerative is the generic name. So.
Okay. Thank you. Thanks very much. And Dr. Park, can you tell us about what you do at UC Davis and maybe just give a quick intro to your bone marrow stem cell trials?
Thank you. And thank you to the organizers of the symposium for including me and the panelists. I am an ophthalmologist who takes care of patients with retinal problems. So I am a fellowship trained vitreoretinal specialist. I am also a full-time faculty member in the Department of Ophthalmology at UC Davis. And as such, I do teaching as well as research. And because I'm a clinician seeing patients, I do clinical research, but I do have a PhD and I do conduct bench or laboratory research. And my area of interest is exploring and developing new treatments for blinding retinal conditions that my patients have. And of interest, I'd like to take observations that we make in laboratory research and bring it to clinical trial to see if these treatments work for our patients with untreatable vision loss from retinal conditions. So the area of research I wanted to share with you is the use of stem cells in our own bone marrow as potential treatment for blinding retinal conditions. Now, Ben did a nice overview of various stem cells that are being used for tissue replacements. So basically you're making pluripotent stem cells and then genetically modifying them to become retinal cells so you can replace the damaged retina in patients with retinal problems. We're taking a slightly different approach. We are exploring the repair potential of stem cells that we have in our own bone marrow. And it's been known now for over 20 years that there are stem cells in our bone marrow characterized by a protein on the surface called CD34. And these stem cells are repair stem cells in our own body. So if we have a damaged tissue, including damaged retina, the damaged tissue releases a signal into your bloodstream. And the signal mobilizes these repair stem cells into your circulation. And the stem cells find the damaged tissue, if it's retina, and incorporate into the damaged tissue and secrete factors, growth factors, proteins that stimulate repair. So we wanted to see if we can maximize the repair potential of these repair stem cells we have in our bone marrow. And we have, and others have shown that you can harvest these stem cells and inject them directly into the eye, similar to the way you get injections for wet macular degeneration or diabetic retinopathy. And we've shown in animal studies that these cells incorporate into the damaged tissue and cause molecular changes in the retina that seem to stimulate repair. And we have shown in animal models of diabetic retinopathy that you can preserve the retinal vascular damage associated with that. We've also seen in an animal model of retinal degeneration that you can preserve the retinal function and retinal cells that are damaged from that condition. So what's nice about this cell therapy is that it's not unique to a specific disease or to specific cell. The effect of the stem cells on the damaged tissue appears to be a response to whatever the damage is, and it tries to reprogram the cells to repair themselves to whatever is causing the injury. So in a sense, it has multiple possible repair roles and may potentially have broad clinical applications. So what we've shown in animal studies is that you can inject human CD34 cells from bone marrow into animal eyes with immunosuppression, so you don't have a cell rejection, and there's no bad thing that happens in the eye or in other parts of the body of the animals. So there seems to be long-term tolerance of the cell therapy. And based on the animal studies, we did get clearance from the Food and Drug Administration to explore the cell therapy in patients with blinding retinal conditions. And we felt that this cell therapy can be applied for different types of diseases, whether they be retinal degenerative conditions like retinitis pigmentosa, whether it be circulatory problems like diabetic retinopathy or retinal vein occlusion. So we took all comers. And for the pilot study, basically it's a safety study to see if we can do this and whether there are any bad things happening in the eye with this treatment. And we noted that this treatment is feasible. We can harvest a large amount of cells for injection. And so far we haven't had any negative effect of injecting the cells in the eye. And more than half the patients seem to have some visual gain that we can note both subjectively and objectively. And so based on these observations, we are in the process of exploring this cell therapy for specific clinical indications. So in our early phase study, the patient who seemed to have the most dramatic improvement in vision was actually a patient with central retinal vein occlusion. We could see changes in the retina as well as visual acuity changes. And so currently we were very fortunate to receive a grant from the National Eye Institute. National Eye Institute is part of the National Institute of Health, which is the big federal agency that funds most biomedical research. And we are conducting a phase 1/2 study exploring the safety and potential efficacy of this cell therapy in patients with persistent vision loss from central retinal vein occlusion. We are also getting more animal data and clinical data in patients with retinitis pigmentosa, and we are in the process of looking for grant funding to explore this cell therapy in retinitis pigmentosa, a phase 1/2 study. So that's where we are right now.
Great, thanks, Dr. Park. And I want to come back to you about a particular phrase you used, that you got FDA clearance. That's a really important aspect of your study that a lot of stem cell studies don't have. And we'll come back to that. But Dr. Rodman, do you want to tell us a bit about your role at ProQR?
Well, I'm glad to. Can everybody hear me okay? Oh, that was a rhetorical question, huh?
You're coming in a little faintly. Maybe if you can move closer to your mic.
Okay, here we go. Better?
I think that's better.
Okay, folks, I hope you can hear me. So I'm Dave Rodman. I'm a physician by training, and at ProQR, where I lead R&D in eye disease, I always found it ironic because I'm trained as a lung doctor and a critical care physician. And so finally, with COVID-19 complicating everything, I feel like my training is coming back into vogue. And so I noticed, for instance, there are a lot of questions that people have about how it's going to affect trials and so on. And feel free to ask those questions and maybe I can answer them in a little while. So at ProCure, we've got a platform technology which is really just a little bit different from gene therapy and gene correction, even cell-based therapy. And that's, we produce these little oligonucleotides that are similar to the ones your body makes. And they, because of that, the cells in your retina will take them up, thinking that it's a signal they're supposed to look at. And so we can inject our drug into the posterior part of the eye, but we don't need to do surgery. It's a simple outpatient procedure. And every cell in the retina takes it up. And what that means is that in contrast to a gene therapy, which can be terrific for changing the biology in a small part of the retina, our drugs can affect the entire retina. And that's really good in early disease. So our theory is that our drugs can go into patients, babies and patients as soon as they become symptomatic, and ideally prevent progression and the need for gene therapy later in life. So I'll stop there and answer questions later when we have time.
That's great. Thanks, Dr. Rodman. So let's go back to Dan. And I have a two-part question, and you can take your time with this one. A lot of people are always interested in participating in clinical trials, and it's not a trivial thing. And can you talk about what people should expect when they enroll in a clinical trial? And then part two is obviously COVID-19, obviously and unfortunately, COVID-19 is having an impact on clinical research. And if you could talk about that secondarily.
Yeah, so why don't I take your second question first? That's a shorter answer. Right now for our group, we have temporarily suspended all patients visiting the hospital trial centers in light of all the recent circumstances with COVID-19. Patient safety is obviously paramount. So at this point, for our group, we have not continued those clinical trials in person. Now, some of the clinical trials that we're doing, long-term follow-up studies and things of that nature that can be done by phone, those are continuing. However, any in-person visits for the trial are currently suspended. When it comes to our treatment centers, and I will just mention that those are really the hospital's decisions, but for the most part, only the most medically necessary visits are being conducted at those centers, and those are really at the discretion, obviously, of those hospitals. So when it comes to clinical trials, this, especially for gene therapy, and we're talking about gene therapy trials that are specific for a certain gene. So many of the things that Ben talked about were concerning what we call specific gene therapy trials, whether it's X-linked RP for RPGR or some of the achromatopsias. So first and foremost is really that you have to qualify. So there's a certain amount of criteria that you have to fulfill. One of the most important is obviously genetic testing. And even if you're not thinking about being a part of a clinical trial, if you have an inherited retinal disease, obviously the definitive diagnosis is to get your genetic test done so they can confirm which gene is having that variant or mutation. So with that said, clinical trials are a little different in that we really are looking for a subset of the population. And so there are certain criteria that you would have to be a part of, certain aspects of maybe your vision and acuity or visual fields, or in our case, because we were using a mobility test, you had to have a certain level of performance on that test so we could measure any kind of change in visual behavior after intervention. With that said, clinical trials take a lot of time. They are usually inpatient visits. Obviously, for our case, it meant that you would have surgery in one eye for delivering of the gene in our phase 1 studies, and then in both eyes in our phase 3 studies. And so that obviously involves hospital time, it involves traveling to certain centers. For us, we had 2 centers in the United States, and all the surgeries were done at those 2 centers. as well as all the follow-up visits were also done at those 2 centers. And for some of our patients, that meant crossing the ocean. And these visits can be relatively frequent in the beginning. And so there is quite a bit of time involved. And as well as when you're actually at the center, there are a number of tests that we would want to do. Obviously, we try and keep in mind that you only have so much energy and so much time to do these, and so we space it out. And because our clinical trials involve children, we also had to keep that in mind, and we space things out a little more. You'll find clinicaltrials.gov is a great resource that it lists clinical trials around the world. But I will put a caveat on that, is that it doesn't mean that it's necessarily gone through any kind of vetting process. And so you have to be careful about clinical trials because some of them, well, I'll just say what for us, there was no cost to the patient, obviously, no cost for the drug. They received the quality care throughout the whole process. We paid for all the travel and expenses and things of that nature. And that's the way clinical trials should be run. It really should not be any cost to the patient, because the patient is already putting out a lot of time and effort and really saying that I don't know exactly what's going to happen, but I'm willing to be a part of your trial. And obviously there's an extensive informed consent process that is very important, where the primary investigator will talk to you about all the potential benefits that might be, but really what the risks are. And because our procedure actually requires a surgical procedure, a vitrectomy, and a subretinal injection, it's not only the risk that there might be with the actual virus and the transgene that we are now using to do gene therapy, but also with the surgery itself. And that's just whether it's part of gene therapy or not, they would go through that. So they want to really talk you through the whole process, and you really should be in a place where you are comfortable, your questions have all been answered, to make that kind of a decision to move forward in a clinical trial. So those are the basics of most clinical trials. Phase 1 clinical trials are usually more about safety, and they may even do different doses to try and figure out which dose is the best and most effective while still being safe. And that's really paramount in all the trials, whether it's a phase 1, 2, or 3. The safety profile is definitely one of the main things that they're looking at. And as clinical trials go, the early phases are really more about safety. So a lot of times, especially in these rare diseases, you'll see a phase 1, 2, where they combine things of that nature. But it's really phase 3 where they will say there's a primary outcome measure. So one test that really wants to drive home the therapeutic benefit of the intervention. Although they look at the totality of the data. So they'll look at everything, and there'll be a primary endpoint, and there'll be several secondaries and possibly some exploratory and tertiary endpoints as well. But in phase 3 is where they really look to see if the patient has benefit, and really it is all about changing the quality of life for that patient. Is it clinically meaningful at the end? So that's the main thing on a clinical trial.
That's great. Thanks, Dan. So Dr. Park, there are a lot of alleged stem cell clinical trials or stem cell therapies, as Dan was alluding to, that people can pay for in clinics. Some are in Florida. For some reason, they're in the South, and they tend to be overseas as well. And often they're taking adipose tissue, fat tissue, or sometimes bone marrow. But those trials aren't really trials, because as Dan said, people are paying for them. And even more important is that the FDA is not monitoring those trials for safety. And can you talk more about the FDA's role in authorizing a clinical trial and what they require from you to ensure that what you're doing is as safe as possible?
So thank you. This is a very important point. As I mentioned, we have been in conversation with the FDA. In fact, we talked to the FDA 3 years before we even initiated the clinical trial to find out what they exactly needed to show that the cell therapy is safe enough to explore in patients. And they gave me a list of studies I had to do in animals first. And it took us 3 years to finish that study before we got approval from the FDA to test the cell therapy in patients. Now, we did publish the preliminary findings of the first 6 patients we treated. And unfortunately, people have taken that data as justification to treat patients, often very desperate to get some visual gain, charge money, and inject cellular material from bone or fat tissue that have not been characterized. And these things are being done without any regulation from the FDA or IRB. They're often charging a lot of money to the patients, and often they're treating both eyes, which is really kind of dangerous. We never do that in regulated clinical trials because you worry about possible bad things happening from treatments that have not been fully characterized and still investigational. So, so currently, I don't have a map to show you, but there are, quote, stem cell centers popping up all over the US and international. And many of these centers are also saying they're treating patients with vision loss from macular degeneration or other conditions that we cannot treat currently. And they're charging thousands of dollars, treating both eyes. And the New England Journal of Medicine journal recently published case reports of patients who lost what remaining vision they had in both eyes from trying these treatments. And so it's very important to know that currently, except for the gene therapy for RP65, there is no gene therapy or stem cell therapy that's been FDA approved. All the other treatments we're discussing today are all investigational, meaning we're still trying to figure out whether the treatment is safe and beneficial to the patients. And so just to the attendees, just be very careful of any, quote, clinical trials, even if they're registered in clinicaltrials.gov, if they're treating both eyes or if they're charging a lot of money. And if you have any questions, you should check and see if they have clearance from the Food and Drug Administration, because they're very particular about trials that they clear to for investigation. They have very high standards, and they want to make sure that we have maximum data on safety and efficacy before patients are subjected to these investigational treatments.
That's great, thank you. Yes, the 2 takeaways: don't pay, and just because it's on clinicaltrials.gov doesn't mean it's a legitimate trial. So very good. And Dr. Rodman, can you talk more about how your antisense oligonucleotides work? I know I gave kind of a layman's or poor man's discussion of how the antisense oligonucleotide works, and maybe starting off with what RNA are might help with that discussion as well.
Okay, Ben, let me give a try at that. I'm going to, I don't want to get too technical, but when we look at the first 3 programs we have that are all in the clinic right now, the first program was for a disease called Leber's congenital amaurosis type 10. And the type of mutation, so that's inherited as an autosomal recessive. So both parents give a defective copy of the gene. And the mutation we're after, which is the most common mutation in LCA, is what's called intronic. And what it does is it makes the cell have a mistake when it goes to take the gene and turn it into the template called RNA, which is in between making the gene and making the functional protein, it puts a typographical error in there that leads to the protein not being made. The second disease we're studying is called Usher syndrome type 2A. Again, a relatively common mutation in that disease we're targeting, but what we target there is a hot spot in the gene in a coded region, something called exon 13, where dozens of mutations can occur. Again, you need to inherit one copy from each parent, but it doesn't have to be the same mutation. It just has to be in that place to get in our trial. Patients can have mutations in other parts of the gene, but as long as they have one copy in exon 13, we can go after it. So there, the RNA, we instead of in the first case where we essentially put a patch over the mistake so that the cell makes a normal protein, in the second disease, we actually chop out a tiny little piece of the protein itself at the RNA level, and the protein that's made still works, even though it's missing that little piece. And finally, the last disease, which is dominant negative retinitis pigmentosa, here you only need to inherit one bad gene from either parent, and it will overwhelm the normal gene in the other copy. And so here what we do is we simply try to suppress or knock down that bad gene and let the good gene come out and work. So we're looking at 3 different mechanisms, but only one basic technology, which is in a computer program, identifying and then designing drugs that can affect what we want to do, either mask a typographical error, cut out an area of mutation, or suppress completely a defective gene, and then move on then to see if it's therapeutically efficacious. So it's a very flexible platform. It's highly computationally based. So we, as opposed to screening hundreds, thousands, or even millions of molecules, we screen about 30, and we rapidly get down to a panel of 3 or 4. And those are the ones we put into the retina from patients that we've grown in the lab. Take the best one of those and then take it into the trial.
Great, thank you for that explanation. I'm going to ask I have one follow-up question. And the ProQR approach involves an injection into the vitreous of the eye, the middle of the eye, which is different, at least today, than where a lot of other treatments get injected, which is subretinally. Can you talk about the benefits of intravitreal injections?
Well, sure I can, Ben. And let me just say that I'm a gene therapy guy, and I worked in gene therapy, and I think gene therapy is a really powerful tool. And I want to talk about the benefits of gene therapy just for a minute before I answer your question directly.
Sure.
So when you look at a disease where there might be 100 mutations that cause the gene, and ultimately the defect is you don't make enough of that protein, gene therapy is really the most rational approach to fix that, because you don't have to be specific to the mutation. You just need to put in a new copy. The challenge is that the toolkit we have today for gene therapy is still pretty rudimentary. And remember, we've been working on this since the 1990s. I used to do therapy for cystic fibrosis, and I've done human trials for CF with gene therapy. The body doesn't particularly like viral vectors. The viral vectors themselves can't be used twice usually because the immune system will recognize them the second time. They have, right now, the delivery is somewhat limited in terms of how much of the retina you can treat. It takes surgery, and the size of the genes we can put in are relatively limited. All of that will go away with gene therapy. We will end up with vectors you can put in the vitreous that get to all the cells in the retina. We will end up with vectors that can handle a bigger expression cassette. And we will end up in a situation where modest immunosuppression allows us to redose the drugs. It's just we're not there today. And so when we looked at the technology that was available with antisense and other oligonucleotides, what we realized was the eye is a very specialized compartment where oligonucleotide therapies are ideal. And so what we find is that if we inject our drugs into the vitreous of the eye, not only don't they get blocked by this limiting membrane and get to every cell, but we make them very stable. So we only have to dose the drug once a year or once every 6 months. And we can redose, and it's pretty simple, and the complication rate's quite low for the procedure. And so those are the advantages. In this day and age, in 2020, it's really the only technology that's available that can get to all the cells, can have a pretty durable response, and wouldn't preclude patients in the future getting a vector-based gene therapy should that turn out to be the more appropriate treatment for them down the road. So it has a lot of advantages, and the biggest one is it's here today, and it can be used today, and it's already been used to make drugs as well.
Thank you, Dr. Rodman. And yes, so far, your emerging therapies are performing impressively. Chris, I think we're ready to open it up to our attendees and try to field some questions from them.
Sure. So there's many questions, and just so everybody knows, we are going to try to address as many questions as we can today. If not, we will reach out directly to you to address those questions. Questions can be asked through the chat and also the Q&A box, and you can also send questions to info@fightingblindness.org. Ben, there's lots of questions coming in about clinical trials. Can you remind people, because they are getting very specific to certain diseases, can you remind everyone what's the best approach someone can take to learn more about the clinical trial status for a particular diagnosis?
Sure. Really, the simplest thing is to go to clinicaltrials.gov, and you can search on diseases. The information can be a little overwhelming because there's a lot of information, but it will tell you what sites the trials are at. You can also go to fightingblindness.org, our site. We do report on many of the clinical trials, and we do have our handy dandy clinical trial chart. You know, maybe that's something we can send out to people. And you can contact us as well. I mean, it does take a little effort to figure out what trials are right for people, depending on what their genetic profile is, where their vision is at, if they have a lot of vision left, if they have more advanced disease. But I think starting at clinicaltrials.gov and fightingblindness.org are good places. And depending on your eye doctor, some eye doctors are really good in this space and they know the trials. Others may not be quite as tuned in. You may want to reach out to your doctor as well.
Thanks, Ben. Another question that was also coming in, the benefits of my Retina Tracker. Should someone, once they're registered, should they just sit around and wait? Or is it one of those that they will be contacted if they may qualify for a clinical trial?
Good question. So I would never just sit around and wait. First of all, if they have new information about their disease, if their vision changes, they find their gene, upload the information. Secondly, and this doesn't address the question directly, but just by being in my Retina Tracker, your data may be observed by researchers and clinicians, even if you're not notified. So let's say a company goes in today, but they're not planning on launching a clinical trial for 3 years. But if they're looking, and I'm just being completely hypothetical, but let's say they're looking for people with rhodopsin mutations, and you're in there and you happen to have a rhodopsin mutation, and when they do their search, they get a lot of people that hit their search, and they never get your personal information, but they get a lot of hits. then that tells them that they can do recruitment in a few years for a clinical trial and get the people they need. So it's important to be in now so you're on the radar screen of companies that are looking off into the future for clinical trial recruitment. In the meantime, as I just said, go to clinicaltrials.gov, talk to us, visit fightingblindness.org. And obviously, this very webinar was, I have to say, a good way to learn about clinical trials that are underway.
Thanks, Ben. One more question for you before we go to our panelists. If someone has been genetically tested in the past, do they qualify for the open access genetic testing? Or also on the flip side, if a gene has not been identified, are they eligible?
Sure. So the panel that we're using, or that Blueprint Genetics is using to get no-cost genetic testing of that panel, you can't have been tested with a larger panel that's usually 32 genes or more since 2016. Because if you have, then you don't qualify for our panel. But if you got results that you're not quite sure what to do with, perhaps they're inconclusive, perhaps you don't understand them, I think a really great exercise or thing to do is to contact Inform DNA and get genetic counseling, because they'll be able to look at your report and guide you in what the next best steps are. Their genetic counseling is available over the phone. They're extremely knowledgeable about our space, the IRDs. If you get genetically tested through our program, you get Informed DNA genetic counseling at no cost. If you're going directly to them with a report you already have, there is a cost, but it may be covered by insurance. Regardless, I strongly suggest people talk to Inform DNA. That really helps people understand what those results mean and what they can potentially do down the road. Sometimes it makes sense to have other family members tested. There are a lot of potential paths, and Informed DNA will help people understand what the best ones are.
Thanks, Ben. This question came in for Dr. Rodman. Do you have the capabilities to test in your facility for autosomal dominant RP1? And if so, how do we find out more about that and getting in contact with you?
Well, I'd say the best way to is to get a hold of me, and I'll answer that. I'll talk to my team to get a hold of them. We don't do the genotyping though, so we would recommend that you go through the FFB to get genotyping done. But I certainly can be helpful to you if you want, and the FFB can give you contact information if you reach out to them.
Yeah, I would say reach out to us on that. I have some more questions about what this person means by testing for RP1. It sounds like they may know or have a sense of what the gene might be. So I want to make sure I understand exactly what they're trying to figure out.
Sounds good.
Thanks. In broad, this is overall for all the panelists, and I know Dr. Chung, you spoke to this a little bit. One of the questions is, with the coronavirus and COVID-19, how is that impacting, further clinical trials or even treatments that are undergoing right now?
Well, I guess I can start with that. For us, the treatment that involves actually pre-treating patients with steroids, which dampens the immune response, because anytime you do any type of intervention in the eye, you tend to get some type of inflammatory response. Usually it's very mild, but because of that, the hospitals have postponed all those treatments as of now. And again, these are Hospitals, they make those decisions as to what they believe to be best for the patient. Obviously, we fully support that. And again, when it comes to our clinical trials, we have informed our ocular centers, and I'm talking about eyes, but I believe it's company-wide. We have postponed any further intervention or in-person visits to our trial centers.
Great. Is there anyone, Dr. Rodman or Dr. Park, would you like to address that from your experience right now?
Well, I'm sorry, I was responding to some of the questions. I didn't hear the question that you're discussing.
Sure. Basically, for each of the panelists, some of the questions that have been going in is, what is happening within the space from treatment to diagnosis to clinical trials with coronavirus and COVID-19?
So, you know, basically for all medical centers with shelter-in-place restrictions from the government, we are limiting care to patients who need urgent care. So for clinical trial, including our clinical trial, as was mentioned by Dr. Chung, we are limiting patient visits unless there are safety issues. So we do contact our patients by phone, and if there are any concerns that there might be some safety issues, then we would have to have them come in to make sure that they're properly taken care of. But as a practicing ophthalmologist, I only see patients that need urgent or intervention. And so we are not seeing patients for routine testing and routine examination. So I believe that's the case for most clinical trials at the current time.
Yeah, so I can answer for ProCure. For the most part, we're in the same position. We have multi-center trials that are global going on, and there's quite a bit of heterogeneity in terms of where local regulations preclude patients from coming in for almost any reason versus sites where, say, the ophthalmology group is so separated, say, from the general hospital and the critical care facilities that, in fact, they can rely more on just local regulations. And so out of maybe 20 sites globally, we do have one site that so far it wants to continue to be active, and their local IRB supports that. And so we haven't precluded that. But in general, we're not interested in general in jeopardizing safety by having patients start a trial and then not be able to have their safety visits because rules change. And so we're pretty, we're pretty reluctant to put new patients in the trials, but it's not an absolute.
Great. I see there's a couple questions on the phone. Jim English, you're able to unmute yourself to ask the question. All right, we will go to the next person. Gary Asano, you have the ability to ask the question. You can unmute yourself. Go back to Jim English. See if you're able to ask your question. Sorry about that. We are moving to Stephanie Anderson. Looks like you have a question that you would like to ask.
Yes. Yes, I do. Can you hear me okay?
We can.
Oh, good. I have RP, and I have a significant part in my left eye that's gone. I live very rural area where there's 30 miles from town, completely off grid. So it's hard for me to get to a town of anything. Okay. Dr. Heckenlively, is that his name? Heckenlively? Okay.
Yes.
He's the one that diagnosed me at UCLA many years ago when I was about 24.
Clearly.
And anyways, my vision's going bad, really bad. I noticed 3 days ago, trying to put my makeup on, I saw a photo of me in the mirror, and I couldn't get my eyes to focus at all, and it's kind of got me scared. And I would like to know what's the best route that I can take. Any suggestions that I can get from you guys? Because I have to travel like 200 miles just to see a doctor for my eyes. And the one I was seeing in Ridgecrest, I'm in California, the one I was seeing in Ridgecrest is now no longer there. So I was going to go down to like Huntington Beach area, Anaheim area. My daughter's in Carlsbad. So I'm going to be quiet and listen to what you guys have to say, please.
This is Ben. If you want to reach out to us, we can see if we can find a doctor that's most convenient to you to see. I think that's, getting to an eye doctor is an important thing. We can help you potentially find some options. I realize nothing is close, but from a relative sense, is UCLA fairly close?
Yes. Like I said, my daughter lives in Carlsbad, so that would be a problem of getting there, because I no longer drive for the safety of others and myself.
Sure.
I've taken that upon myself, that I know I don't have any peripheral vision at all. My left eye's gone. My right eye's getting worse. So I don't want to take a chance. But my daughter's more than willing to step up to the plate and help me and be with me. So yes, I can get to UCLA.
And they have some physicians there, eye doctors, who are expert in IRDs. Obviously, with the COVID-19 situation, I would suggest you emphasize the urgency, and maybe you can get in, but that could be tough.
You know, stress. I heard stress has a lot to do with my vision deteriorating quickly. Is that?
You're going to have to pass this question on. This is.
I think we're losing Stephanie.
I'm right here.
Okay.
Yeah.
You know what, Stephanie? What I would say is let's talk further.
Okay.
We can talk offline, and I'll see if we can connect you with somebody, and get you connected with the right doctor.
Okay, that would be great. Awesome. Yeah, I am registered with the Retina Tracker and Foundation for Fighting Blindness and all that. I am registered for all of that. So
great.
Yes. Oh, that would be awesome. Do I just wait for an email, or you guys will get a hold of me? You have my email address.
Yes.
We'll
figure it
all
out.
of you for all that you do. Thank you so very much from the bottom of my heart. I know that, I'm very grateful. So I wanted to give you kudos for that.
Thank you.
Thank you.
I think we have probably time for another question or two. Dana Simon, you have been selected, so you're able to unmute yourself to ask your question. Dana Simon.
Hey, Chris, where's the unmute button for most people? I know I'm looking at a full screen. It's in my bottom left corner. Maybe it would be helpful to mention that.
Yeah, Stephanie Anderson, you should be good to go now.
Okay.
Thank you so much. Thank you.
Hello, this is Dana Simon.
Dana, we hear you. You may go ahead.
It took me a while to figure out how to unmute. So, yes, my question is these therapies, are they better for people with more advanced ushers or RP that are like 2180 or higher, or are they for people with like 2070 or 20, because in that case, what other, if there's also the computer chip and other technologies that were not discussed.
Go ahead, Dan. I'm happy to help too.
Sure. Each clinical trial has slightly different parameters as what they are looking for to enroll patients. For our patients, the majority of them were probably in that 2060, 2070 range, and all the way up to counting fingers or hand motion vision. So there is a relatively large range. So when you talk about the chip or some of these other therapeutics that have been mentioned there, so gene therapy, you have to have cells left. So there has to be some structure left for the viral component to actually deliver the DNA. So if you have no cells, there's nothing for that vector to actually target. So some of the things that they have looked at, you have mentioned the chip, there's optogenetics, and potentially some stem cells as well, look to recreate that cellular level or that function that has been lost because there is no RPE or photoreceptors that are left in the amount needed. So for gene therapy, though, you do have to have some cells. So it probably would not be something that if you had very little light perception vision or no light perception vision, gene therapy may not be the best thing in its present state. Now that could change, and every case is on a case-by-case basis now that the treatment for RP65 is in the commercial realm and not in a clinical trial realm. So I'll just put those 2 cents in to start.
Okay. But I just want to add that I do have light perceptor, can see colors, but in 2013, I was able to read with glasses, but I had a drop in my vision, so.
Yeah, this is Ben, and I just want to emphasize what Dan said, is everybody's case is a little different. The state of everybody's retinas is a little different. And some therapies may work better for people with less vision. Others may work better for people with more vision. So it's good to look at the criteria for the clinical trial on clinicaltrials.gov. And then ultimately your doctor can help you understand if your retina is in a situation that it can potentially benefit from a particular approach.
Great, thanks, Dana. Ben, one more question before we go. This was from Gary. He wasn't able to get the mic working, but his question was, you know, with so many people out there and they still have some functional deficits, even with improved visual acuity, is it essential that low vision rehabilitation be incorporated, at least for post-rehab? Or otherwise, with the sparse number of low vision practitioners, would it be a haphazard search for those treated to find them? And what do you think?
Well, I will start off by saying I am not a low vision rehab expert, but I would say in general, I think it's good to try to talk to a low vision expert, whether it's a rehabilitation person or optometrist in your community, and see if they can help you. I think they can offer a lot of value to people. And again, I would just say in general, I think it's okay to pursue something if you think you might benefit from some type of accommodation. I don't know if anybody wants to add to that.
I agree. For any patient who's not happy with the vision with the best corrective glasses, to seek low vision evaluation, just in case tools or techniques that they can train or offer might help you maximize the vision that you have.
And one other comment, if you send us an email, or Chris, maybe you already have that information, we can potentially connect you with a group that can help you find a good low vision rehab person.
Yeah, absolutely. And Ben, thanks for all the questions. The panel, Dr. Chung, Dr. Park, and Dr. Rodman, thank you for your time. For all the questions that have come in, unfortunately, we aren't able to get to every single one of them, but we have captured all the questions. We will have them each addressed individually and contact everyone individually. So we will have answers to you. So I want to be able to hand that back over to Donna Cohen.
Participating in today's webinar. Before you leave, we just want to announce the official launch of our National Virtual Vision Walk Day that is happening on Saturday, June 6th. For more information and details, please reach out to your local foundation office. Our Vision Walk may look different this year, but our mission to fund sight-saving research is unchanging. Our next webinar will be held on Saturday, April 18th from 10 a.m. to 12 noon Eastern Time. Speakers include Ben Shaverman, Daniel Chung, Jill Dolgen of AGTC, and Gohar Salam of Retina Institute of Indiana. On behalf of the Foundation Fighting Blindness, we want to thank our speakers today, Ben Shaverman, Daniel Chung, Dr. Susanna Soon-Chun Park, and Dr. David Rodman. Just a reminder, if you have any additional questions that were not addressed or you did not send in today's session, please email info@fightingblindness.org. And finally, we want to thank every one of our attendees for joining us on today's webinar. The Foundation is a beacon of hope, and because of your support, we are able to shine even brighter. So please, everyone, stay safe, and we hope to see you on our next webinar on April 18th. And this concludes today's session. Thank you all.
Thank you.
Everyone, stay well.
You too. Thanks. Have a good day.
Thank you.